Okay, let me share my screen here. And start up my slides. Okay, so first I want to say to Carl and David, thank you for organizing this. I see a lot of familiar faces on the screen. I see a lot of new faces. I know some of you have torn yourself away from the election. And so I want to promise you an anxiety-free time here. Carl tried to up the anxiety by making it sound like I was going to do a lot of math, but I'm not. And so I hope you just enjoy a little respite from the anxiety. And the other thing, in contrast to the election, I promise to reach a conclusion in my time. So I'm going to talk about the fly. And in particular, I'm going to ask the question, sorry, this is not, yeah, there it goes. How does a fly know which way it's traveling? Now, you might think that's an easy question and just say, well, that way. The direction that the fly is heading, we call the heading direction or facing. But a fly travels around in wind and often is not going in the same direction that it's facing. It's drifting to the side due to this wind. And therefore it needs to, if it wants to know, where it's really going in the world, needs to compensate for this. And in its navigational system, figure out where it's actually traveling. So we distinguish between heading direction, which is what I've shown, and traveling direction, which is the velocity vector of this fly. So that would be, in this case, something like that, the traveling direction. Now, flies have ways of determining that they're drifting or which way they're going. In the visual system, there are various optic flow sensors that project the direction of motion onto these vectors that I've shown. And so, that's the kind of calculation I'm going to try to tell you about. But those vectors, of course, are attached to the fly. They're part of the fly visual system. But if you want to get to a specific target in the world, or you want to know, what path did you fly on, the fly needs to sort of anchor these vectors to the world around. So there has to be a transformation from allocentric, or the sort of egocentric, rather, coordinates of the fly into allocentric world coordinates. So we'll talk about that too. So this is a problem that is in representation. How are these angles represented in the brain of the fly? But it's also in computation. Traveling direction has to be computed. And that involves vector addition of these red vectors, or the motion projected onto those red vectors. And it also requires a coordinate transformation from the coordinates of the fly's body to the coordinates of the world around. So I'm going to tell you in great detail how this works in the fly. And the reason that I can do that is because of these two guys. So this is work done in collaboration with Gabby Mayman and Chung Liu, who's a graduate student with Gabby. They did the experimental work, but they also really provided the insight of working the way through a complicated circuit. I'll try to stress that as I go along. And really, I just multiply cosines every once in a while. That's my role in the collaboration. So I'm really describing their work, and I can say it's their very beautiful work. Okay, so this last picture is just to remind me that flies are not the only animals that travel sideways or at an angle sometimes. Of course, every animal does this and has to compensate for the fact that at times we don't move exactly following our noses. Okay, so let me start off with heading direction. Some of you may know this very beautiful work from the Jayaraman lab. Johannes Sillig and Vivek Jayaraman discovered a system in the fly that tracks its heading relative to the surrounding world. The second paper that I cited here has a very nice model of this too. So there are really elegant models and a pretty good understanding of this first step. How is heading represented in the fly? And that occurs in this donut-shaped region of the brain that you see in the picture called the ellipsoid body. So that's the first player in our journey here. And what Sillig and Jayaraman discovered was that there was a hotspot of activity in the neurons that innervate this ring and that hotspot activity, like a compass needle, will track fixed objects in the world as the fly turns. So it's providing the heading direction in a world coordinate system. Here's just an example of that from work of Sung-Soo Kim in Vivek Jayaraman's lab. So this is a picture of a forest scene. It's been reduced in resolution down to kind of fly level resolution, and then shown to a fly in a virtual reality environment where the imaging of this bump of activity in the ellipsoid body could be done. And here you can see that activity is locked onto a particular angle. But the key is that if you shift this scene around the fly like that, the bolus of activity shifts in exactly a corresponding angle. And you can see that here, where in blue, this is a tracing of where in the ring, ellipsoid body ring, the bump of activity was. And then the red is just tracking some arbitrary location in this picture. And you can see they track each other really quite well. So that's the fly's compass system. It looks, in a schematic like this. The fly turns, but the compass system keeps pointing towards an object. Now, you might realize here that as the fly turns here to the left, the fixed object effectively turns to the right. So there's a minus sign here that's going to crop up from now and again. Just this is an anxiety-free lecture, so don't worry about it, but it'll come up. But nevertheless, this system represents the heading of the fly. Now, there's another player in this system. Basically, what I'm doing here is introducing you to various pieces of what's called the central complex in the fly. This new piece is called the Protocerebral Bridge, or just the bridge, I'll probably call it. It's another player in this heading representation. And it works like this. First of all, these regions are divided into segments, into compartments, if you want. And those compartments are innervated by different members of this class of cells. So it sort of looks like this. The ellipsoid body ring is down at the bottom and the protocerebral bridge at the top. And what happens is that the bump of activity that I've shown you is in neurons called EPGs. These letters stand for where this neuron innervates. It stands for ellipsoid body, protocerebral bridge, and another region called the gall, which won't come up in my talk. But and so these neurons look like this. They make the bump of activity in the ellipsoid body that tracks the outside world, and then they send that activity up to the bridge, and they do it on both sides. So it ends up that the compass is actually sort of copied into three compasses. And there are sets of cells to innervate each of these compartments and going all the way around the ring. I won't show you the whole set, but going all the way across the protocerebral bridge and all the way around the ring. And they are locked together because they're really the activity of the same cells. And so here from Chung is a recording from Gabby's lab, where you'll see as the fly turns, the activity in these three regions will lock and move all together. So you can see two copies of the bump at the top and the one copy that I've already shown you in the ellipsoid body below. Now, that allows you, those projections allow you to build a coordinate system for this structure. In other words, we can build coordinates going around the ring of the ellipsoid body, just angles, I mean. But because of these projections, you can then assign those angles up into the protocerebral bridge. And it looks like this. As I said, there are two more cycles up there. They're not closed into rings, but nevertheless, they go around the full 360 degrees on the left side and on the right side. And those angles will be very important as we work our way through this circuit. So here's what the EPG neurons do. They take the bump that's created in the ellipsoid body, transfer it up into the bridge. And the bridge is important because that allows this compass direction to be relayed to other cells that then will go on and compute various things. And so the next player in the game that we're going to look at are called PFR cells. That stands for Protocerebral Bridge, fan-shaped body, which is another structure in the central complex, and then round body, which is again a structure I won't talk about. So the PFRs look a lot like the EPGs, but look like they're playing the game between the bridge and this fan-shaped body. And in fact, you know, one way to think, well, they span the different columns and the different regions in the protocerebral bridge in the same way. There are sets of neurons for every compartment. And one thing that does is allow you to take these angle coordinates and reassign them back down in the fan-shaped body. So by looking at how these different cell types connect, we can trace out an analog map of the circle being mapped twice into the protocerebral bridge and then once across the fan-shaped body. So we've got yet another one of these circular structures. Okay, and then the sort of flow pattern that I'm talking about looks like this. In the ellipsoid body, you build the bump at an appropriate angle, depending on your orientation or the fly's orientation in the world. The EPG neurons carry that activity up to the protocerebral bridge. It's transferred to these PFR neurons, and they pull it down into the fan-shaped body. So if you just looked, or here's a better diagram that Chung made that's a little clearer. If you just look at this diagram, you would say, okay, well, the role of these PFR neurons is just to take the compass signal and transfer it from the ellipsoid body where it is formed into the fan-shaped body. And so that really started, that picture started the experiments that were done by Gabby and Chung to look to see if that's true. Is that what the role of these neurons are? Are they just transferring the compass from one place to another? So most of the experiments that I will talk about are done like this. A fly is tethered, but flying, so it can fly along. And the wings are monitored so that by monitoring the wing beat amplitude, they can determine whether the fly is turning. Of course, the fly is not really turning because it's glued in place. But if the fly intends to turn, then this dot that I've shown there moves around appropriately. So this is a closed loop virtual reality that makes the fly sort of think that when it's turning, the world is turning appropriately around it. Okay, and then in this setup, they imaged simultaneously both the compass system in the ellipsoid body, that's what's on the left, and then these PFR neurons that I'm talking about. And often they would find that they were nicely aligned. Remember, we can assign angles the way I showed you to these different regions. So you can say, well, are these two bumps at the same angle? And sometimes they were, but sometimes they weren't. And that's kind of what first alerted them that the PFRs might not simply being a copying system, but might be a little more interesting than that. And so what they did was, you can see these blue dots at the bottom of the virtual reality system. Those are dots that allow them to impose a visual sense of flow in the fly's environment. So the first experiment was just to make an optic flow like this. So it was just as if the fly was flying directly in its heading direction. In other words, zero drift, just flying forward. And they observed, and that's this open loop flow. They observed when that happened, there was a much better locking of these two systems to each other. And that led them to do a systematic set of experiments like this. So what you can see here in the middle is this, optic flow that simulates direct forward flight. But in addition, they could simulate backwards flight, drifting to the left, drifting to the right, et cetera. All these different angles of flight could be simulated. And again, with this simultaneous imaging, they could look at the bump. So in this top blue here, this is the track of the bump as the fly is doing a turn in this virtual reality environment. And here's the track of this second bump, the one that's in the fan-shaped body, the PFR bump. And you can see they're very nicely locked, and between these dashed lines, the optic flow comes on, and they're really very tightly locked here. There's no difference between them. On the other hand, when they simulated backward motion, then when the backward motion was on, these two bumps diverged from each other, and in fact diverged by 180 degrees. And doing all the angles, they found a very systematic relationship between the separation of these bumps and the drift that they were simulating, which, rather than going through every panel here, let me just summarize by saying that the difference between the compass bump and this new PFR bump very accurately tracks the drift angle. Now, if you accept that the EPG angle is the heading direction, and you sort of undo this, what you find is this PFR is a traveling direction bump in the world coordinates. It signals the direction in the world. You might notice here the axis is backwards. This is the sign flip that I warned you about, but told you not to worry about. So traveling direction is represented in these PFR neurons in the same way that the heading direction is represented in these compass neurons or EPG neurons. So obviously the next question is, how does this happen? How is the difference between these computed, presumably on the basis of some kind of flow information, optic flow information being carried by the visual system? So let me just say how we attack this first. So just summarize, there's a bump of activity in the ellipsoid body that tells you heading direction. There's a bump of the activity that represents traveling direction in the fan-shaped body. And we believe there's lots of evidence in the ellipsoid body that is a self-sustaining bump. It's very much like the ring model that's quite famous in theoretical neuroscience. It's a self-sustained bump, and we believe also that in the fan-shaped body, so this bump is also a self-sustained bump, and it can exist anywhere from left to right across the whole extent of this fan-shaped body. So it's a line attractor, basically. Now, in a line attractor, what determines where the bump actually ends up is the input. And so what we focus on then is an input that is going to turn out to be a sinusoidally shaped input that is expressed across this structure, because if we find the max of that, we can say that there's no other influence. This bump can equally exist at any location. So it's going to go to the max of the input. So if we want to understand where this bump is, what we want to do is focus on the inputs to these PFR neurons and determine where they're maximized. Okay, so I told you a little about how this calculation has to go. There's a motion signal that has to come in and be computed to determine drift. That motion signal, comes from the eyes, but it actually comes in an inverted way. So there's an inhibitory pathway. And so actually the motion signal that we're going to be talking about is directed backwards at 45, and it's actually 135 and minus 135 in the backward direction. So that's the motion signal that this system is getting. It has to reference that to an external landmark in order to be in allocentric coordinates, and it has to add the vectors. So that's the calculation we're talking about in order to get this traveling direction vector. The coordinates then, or the vectors we're talking about, are a forward heading direction and two backward angled motion direction signals. And let me just talk about the algorithm if you want, or what's the calculation that has to be done. It looks like this. If the fly is moving forward, then the motion in the backward direction is reduced. So those backward directions get shorter. And if you add up these three vectors, you get a forward signal. So that's traveling forward. If the fly is actually drifting backwards, despite heading forwards, then those backward direction vectors sense more motion in the backward direction. They get longer, and the net vector ends up pointing backwards. If the fly is drifting to the right while it's flying along, then what happens is one of these vectors gets longer, it senses more motion. The other vector gets shorter because it senses less motion. You add them up and you get a rightward vector. And then finally, if the fly simply turns and flies like that, what has to happen is all these vectors have to turn together in the world. And so you end up getting a rightward motion, even though the fly is still flying straight ahead. So that's the calculation that we have to account for. The question is mechanism. How is this done in the fly's central complex? So let me introduce then a way of representing and manipulating vectors that some of you may have learned about in physics class. I actually learned about it in physics class, but I paid absolutely no attention to it and never thought it would come back to actually enter my life. But in two dimensions, obviously, a vector is characterized by an angle and a length. And you can map that onto a sinusoidal wave, if you want, sinusoidal function, by saying that the position of the peak represents the angle of the vector and the amplitude of the sinusoid represents the length of the vector. So this is a mapping from vectors to sine waves. Now, if you have another vector, obviously you can do the same thing, you represent it the same way. And then a nice feature is that if you want to add those vectors, all you have to do is add the sine waves. And the peak phase and the amplitude of the resulting sum will be the length and the angle of the resulting vector. Now, this is used in engineering as a trick to compute how to basically add sinusoidal waves by just adding vectors. But in the fly, it's the other way around. What we're going to see is the fly is representing vectors in terms of sine waves and adding them and shifting them in various ways in order to do vector calculations. That's really the point of my talk, is to convince you that that statement is true. There's another nice trick here that if you want to do a coordinate transformation and, for example, refer to this vector with that other angle, all you got to do is shift the corresponding wave over and you've done the coordinate transformation. So this is a nice way to do vector representation, vector addition, vector rotation, and this has certainly been noticed before. It was proposed for the hippocampus by O'Keefe, where the oscillation was in the time domain. That's not what I'm talking about. I'm talking about an oscillation in space across these structures that I've been talking about. And Turetsky, Reddish, and Wan actually realized that that could also be happening in the hippocampus. So those are theoretical papers. Then it was applied to insect navigation by this, there's a typo there, but Whitman and Schregler paper. And then finally, in a really very beautiful paper, Barbara Webb and Stanley Hines and collaborators applied these ideas to a circuit that's really very similar. It's a circuit in bees, but it's very similar to the circuit I'm going to talk about. I will come back to this paper at the end, but it has a lot of parallels with what I'm going to talk about. Okay, so just to stress, the waves that I'm talking about are spatial waves in a population of neurons across these structures. And we're going to then parameterize them. The x-axis in these plots is going to be either a position angle up in the bridge, if we're talking about activity there, or a position angle in the fan-shaped body. And I showed you how we defined those position angles a little earlier, except for this minus sign. I'm actually going to plot it as a function of minus the position angle. Otherwise, there'll be an irritating minus sign that will crop into the talk. So here's the idea. We want to represent these three vectors and the calculations that they perform in terms of sinusoids in populations of neurons across these structures, for example, across the fan-shaped body. So the first thing is, what neurons are these? Now, the heading direction one, I've already said, that's these EPG neurons. Those are the neurons that represent heading direction. So they're a good candidate for our sinusoidal representation of heading direction. But what about the other two vectors? So, you know, I just push a button on my computer and up come these names, PFNVs. So I want to acknowledge my collaborators, because it was easy for me to pop these names up, but they had to, from the thousands of cells and hundreds of cell types in a central complex, figure out that these were the candidate neurons. I will try to convince you that they're the right neurons, but I just want to acknowledge the insight that went into being able to identify that these are the neurons that carry these other two vectors in their sinusoids. Just to show you a PFN neuron is a good candidate, here's a picture of one. They get a signal in the bridge, that's the bridge up there. So they are linked into the compass system. They give an output down where these PFR neurons are, so they can put their signal into the traveling wave bump that we're talking about. And finally, they get another input in a structure called the nodulus, and that is a visual motion input. So they're good candidates in the sense that they get the visual motion signal and they come from the right place and they go to the right place. But we've got a ways to go to really show that they're correct. So there are a number of conditions that have to be met if this idea is going to get off the ground. First of all, so remember what I'm saying is these vectors are represented by sinusoidal patterns across the spatial structure of these different central complex regions. It's not carried by one neuron, but by a whole population of neurons. And it has to obey the rules of the game. First of all, it has to be sinusoidal, and it has to have the right phases and amplitudes to represent the vectors that I'm showing you on the left. So the first question you would ask is, okay, are they sinusoidal? That better be true, or we're not going to get there. And so here is a picture of Chung took images of PFN activity on the left side and on the right side of the bridge. I've separated them here. And then interpolated them. That's what you're seeing in the brown circles. And then the gray is a sinusoidal fit. So you can see the spatial profile of these cells across the bridge is really pretty sinusoidal. As the fly turns, these sinusoids will shift their phase left, right, but they'll stay sinusoids. So that's pretty good. If you look at the EPGs, the same exact way, you image them across this structure, they're less sinusoidal. They're narrower than a sinusoid, as you see in blue here. But there's an extra cell that carries the compass signal down to these PFR neurons, the traveling direction neurons. And we believe that the sum of those two, which is what these PFRs get, is more sinusoidal. And so I'm going to give us a pass on the sinusoidal activity patterns and move on. All right, the next thing that better be true is that I told you that when the fly moves, for example, backwards I've shown here, these vectors should get longer, and that's how come their sum, ended up pointing in the backward direction. And likewise, I showed you if the fly drifts to the right, the one vector gets long, the other vector gets short. Now remember that the length of the vector is encoded in the amplitude of the sine wave. So if this analogy is going to be true, it better be true that the amplitudes of these sinusoidal activity patterns in the different neurons vary correctly with the motion of the animal, with the drift motion that we're talking about. And so here's an example of that. So again, in the bridge, Chung measured this profile that I've already shown you this, but now measured it when the fly was experiencing these visual optic flow signals in different directions. And you'll see, and they vary in amplitude, you'll see in a second. So here's the forward direction. But now if you look at the same set of neurons and imaging them when the fly is being simulated in the backward direction, you get a much bigger amplitude of this sinusoidal variation in activity across the bridge. And of course, that corresponds exactly to these two red vectors getting longer in this case. And now you can fill in the whole set of directions. And if I show you various examples, you'll see this makes sense. So for example, this neuron, the left PFN, is encoding this red vector. And so it should be very big when the fly is drifting back and to the right like that. And it should be very short in this case when it's in the opposite direction. On the other hand, the bottom line is showing the representation of this vector. It should be big for motion here, back and to the left, and it should be small. Let's see, which is the opposite one? This one, sorry. It should be small in this case when it's up and to the right. So these amplitudes certainly, look like they're doing the right thing to represent those vectors. But of course, we can just calculate this. I'm for the time being, think of the fly as moving at a constant speed. I'm not going to talk about speed until the end of the talk. So just think about a constant speed. So then you know exactly how these projections should vary. We're projecting the motion of the fly onto these two vectors. When you project, you get a cosine, right? And the cosine should be maximal at these plus and minus 135-degree angles corresponding to the direction of these backward vectors. So this model predicts that the amplitude as a function of the drift angle or the egocentric traveling angle of the fly should follow this cosine. This is a different cosine now, remember. The basic activity pattern is a cosine shape across the structure, but now we're talking about the amplitude of that cosine as a function of the drift angle, and that's another cosine. And you can see the data very beautifully fit this idea. So these vectors really are, when the vectors vary in length, the amplitude of the corresponding sine waves seems to be varying exactly as you would predict. So I'm going to give a victory there, but let me just add one thing, and that is that we're assuming this front heading projection vector is just constant. And so it should not vary as they simulate in the experiments different traveling directions, and that's true. So in the model, we'd expect a straight line, and that's a pretty good fit to the data. So it really looks like these sinusoids are representing three vectors, one at a constant length and the other two varying appropriately with the motion of the fly. So we'll give ourselves a victory then. Okay, the next thing that I told you about when I did the algorithmic view is that if the fly turns, all these vectors should turn together. That's what locks the representation to the external world. So they'd better be locked in phase as the fly turns. And so that's you would expect that to happen actually, because I didn't show before, but in the same way as I showed you the PFRs getting input, the way that the PFN neurons know about the compass signal is they get direct input from the EPGs like this, and likewise as you go around the circle or around the thing. So actually I goofed up on the bottom, but so you would expect this. And again, doing simultaneous imaging of the EPG activity and the PFN, the peak of the sinusoid of the PFN, you can see that as the fly turns in this arena, it has a dot to orient itself to the external world. They track each other very well. It's probably easiest to see if you look at the right plot. So you can see that the difference in phase between these stays really relatively tightly locked to zero. And so you would say, okay, great, the phases do shift together with rotation. We can give ourselves a victory there. But we got one more thing to get right. So the first, are they sinusoids? The second was whether the amplitude is correct. The third is whether they shift together, but they have to also have the right relative phases. In other words, these vectors that we're trying to represent are offset from each other by 135 degrees. Now, according to the rules of the game, that means the phases of the different sinusoids should also be offset by 135 degrees. That's how the direction of the vector is represented. But unfortunately, I just showed you this picture. And this picture shows that they are not offset. They're all lined up with each other. So unfortunately, we've got alignment of these vectors. So what does that mean? Well, we got a lot of things right, but the picture that we would want is what I'm showing here. But the real picture looks like this. They're all aligned with each other, which means that the vectors we're representing so nicely look like that. They're all aligned with each other. So at this point, we'd have to say, well, nice try, but it didn't look like it worked because we failed in this direction thing. But we're not done. Obviously, I'm not done. Otherwise, I wouldn't be talking about this. And the point is that the measurements that we're talking about were all made up in the bridge. And the reason for that is because in the bridge, you can separate left from right. The left cells, are on the left side of the bridge, the right cells are on the right side of the bridge, but they all get mixed up when they go into the fan-shaped body. So what I've been talking to you about is the alignment of these signals when they're in the bridge. Now, the summation that we're talking about, what we're trying to do is identify the inputs to these PFR neurons, and they get added up, not up in the bridge, but down in the fan-shaped body. So we still have some wiggle room. And in fact, if I've been sort of, providing a little drama here by not showing you the connections, but if I now show you the connections of these PFNs to the fan-shaped body, you see something interesting. Here, I've shown you PFNs that connect to these yellow regions in the bridge. But if you notice in the diagram, they're not going to the yellow regions in the fan-shaped body. They're going to the green and the orange. And remember, we've aligned the angles all up in these regions. So in a sense, they're going to the wrong place. They're offset relative to the alignment of angles that I talked about. Again, these are a whole set of neurons, one for every column or set for every column and for every of these what are called glomeruli in the protocerebral bridge, and they're all offset. So if I gave you a summary here, it looks like this. In the left bridge, they flip by 45 degrees to the right, which in my bizarre convention is minus 45 degrees. And in the right bridge, they shift one to the left. So that's good. That means we're getting a phase shift, right? It means that these sine waves look like this. They're aligned. If I showed you them in the bridge, they would all be aligned. But if I now showed them to you in the fan-shaped body, they look like this, shifted by 45 degrees. Well, that's a start, but it's not what we want. That means we're representing vectors that look like this. And so close, but we didn't get there yet. But the next great news is that these PFNs, remember, we're trying to get to these PFR neurons. Those are the traveling direction neurons. They don't go to the PFR neurons. I showed you that they went to the fan-shaped body, but they actually connect onto another set of neurons called H delta B. These are interneurons, and they have the property that they receive their input. This is one of them. Again, there's a whole set of these. They receive their input at one part of the fan-shaped body, but they put their output in a different part of the fan-shaped body. So they are going to provide an additional angular shift. And they are the ones that carry the signal to these traveling direction neurons, the PFRs. So if we take into account the interneurons look like this. Again, there's different sets that are offset and go between two places. If we take them into account, they produce a 180-degree shift. And if you do a little arithmetic, you add up these two angles and bingo, you've got your 135 degrees. So the anatomy here saves the day. And the beauty of this system being laid out so neatly in angle space really allows you to see this. And so we got it. These waves, if you include the interneuron, are offset by 135 degrees, and things are looking pretty good. Let me just show you that what you can do in fly neuroscience these days due to the connectome data that only relatively recently came out. It's publicly available. You can all take a look at it. There will shortly be a paper from Vivek Jayaraman's group analyzing in great detail what the connectome has to say about the central complex. That's not quite out yet, but the data's out. And so we can make use of that. And here's an analysis of that where you can go synapse by synapse and trace through the pathway and compute the shift of every synapse, basically implied by every synapse, and then do these histograms and means. And you can see you really get very close to these answers. At the EM synaptic level, you find that these shifts really are quite accurately in the data. Okay, so I'm going to turn this X into a check mark. We really seem to have satisfied all of these conditions. Just to remind you that these vectors seem to be represented by sinusoidal activity patterns across the these structures. Their amplitudes varies exactly as it needs to vary to represent the different projections of motion. They shift together in phase, which couples them to the external world. Basically, they're all coupled to the compass, and the compass is coupled to the external world. And finally, they have the phase offset to do the vector addition and get the right answer. So here's a kind of a complete model description of the whole system. We can represent these different sinusoids at the bottom. In the middle is this little vector diagram that I showed you, I called algorithm. But at the bottom are the activity patterns that we expect in all these different cases. You can see, for example, the EPG is just a sinusoid center. This is really the EPG plus delta 7. Is centered in the forward direction unless the fly turns and then it shifts over. You can see the right and the left shifted by 135 degrees. They have low amplitude when the fly is moving forward. They have big amplitude when the fly is moving backwards, and they have this, a non-symmetric amplitude when the fly goes right, et cetera, and they all shift together. And then finally, here is what we predict for the input to this traveling direction system. This again is across the fan-shaped body, neurons, PFR neurons that live in the columns at different angles should be getting a profile of input that looks like this. And finally, what I told you is that, to figure out where the bump of PFR activity is, we just have to look for the maximum of that input, because the line attractor should just drift to that maximum. And if you look, the maxima is exactly at the right place. It's at the angle of the purple vectors, which is the traveling direction. So basically, this is the picture of how traveling direction is computed. You can compare the model to the data. The model, as I described it, would get traveling direction absolutely perfect. But if you actually put in the amplitudes that we measure rather than the idealized amplitudes, actually you get really an excellent fit to the data, arguing that, again, this PFR phase is representing the direction. This is done in egocentric coordinates, but is representing the allocentric direction of the fly. Okay, so in a fly, you can do a lot of manipulations to try to convince, provide more evidence that this idea is right. One is, I haven't mentioned much about walking flies, but there's evidence that this system also works in walking flies. One thing that Gabby and Chung did in walking flies was to block synaptic transmission by genetic manipulations in the compass system. So this is a fly that doesn't have a compass, and you would expect that that would detach this traveling direction from the external world. It would still sense motion, but it would be uncoupled from the external world. And that's exactly what you find, is that the phase, which was nicely aligned, so this is in flies, without drift, was nicely aligned to the world before, has now become in this line. This is the manipulated fly, much more flat. So as predicted, this system seems to have decoupled from the external world in that manipulation. Another manipulation is to artificially shrink the length of these backward vectors. That's by silencing the PFNs. Remember, those are the neurons that represent the backward vectors. So if you silence them, it's like you're shrinking the thing. And that should make the fly think it's moving forward. And this is done in those cases where the fly is not getting any visual motion. So in other words, what we would say is that this manipulation is simulating optic flow. And that's what happens, that from a poorly aligned system, you get a much better aligned system, just as if you turned on the optic flow. Finally, you can excite these PFNs. That means you're simulating backwards motion. And indeed, instead of a forward peak, now you get a 180 backwards peak. So the manipulations seem to agree. And so let me just take you through the circuit. You have these EPG neurons representing heading direction. They translate that compass signal to another set of neurons that get visual motion. Those neurons do vector addition. There's this 180-degree correction, and you get the signal going to the PFRs. The PFRs also get the EPG signal, and I don't know if you followed, but basically the only role of that is to do an offset because the zero, there's a baseline activity of these PFNs that would make the fly continually think it's moving backwards if you didn't offset by a forward direction signal. And then finally, the PFRs do a max operation, and out comes this traveling direction signal. Okay, let me just talk a little bit, add a few things. I've talked about this particular system. There are actually more PFN neurons. I've talked about PFNV neurons. There are PFND neurons. If you look at the anatomy, they have the 45-degree shift like these guys, but they don't have the interneuron. And if you've been paying attention, you should immediately be able to know what signal they carry. They have to carry a forward direction signal because they're not 180-degree flipped. And indeed they do. So their amplitude is modulated as if they're carrying a forward direction signal. We haven't talked about them because they seem in flying to have a subdominant role, but they're definitely there in the circuit. And the final thing I want to talk about is path integration. So we've talked about traveling direction. Obviously, that's the relevant direction in an animal that's not moving straight forward. And ego to allocentric transformation, that's what you need to do path integration, vector addition. So we have a lot of the ideas here for path integration. And that was really the topic of this paper I mentioned before, in particular, the really beautiful theoretical work of Barbara Webb, who built these ideas into a really very complete model of path integration. And that, I would urge you to read. It's very elegant. In particular, what it generates is no matter, as an insect moves along, generates a vector pointing back to the home direction. And in animals like ants and bees, that's a very important signal. So the question is this system, what I've talked about? There's evidence that it's not. One is that the speed modulation, of course, I mentioned that I was going to talk about angle and not speed. If you look at the speed modulation of this bump, which is very sensitive to traveling direction angle, it's not very speed modulated. So it's as if speed has been kind of left out in this system, and you can't do path integration without incorporating a speed signal. The other thing is that we don't see evidence for integration. But I would just end by saying, these are the components of what you would need for path integration. The ideas, I think, are generally on the right track. And so, it should be that in the future, this system, the fly central complex, will really offer the community a chance to really work out path integration in a very detailed way. So that's the system I've talked about. I'll wind up now. I want to acknowledge again my collaborators for their really beautiful work, and it's been just a pleasure and a privilege to work with them. I will end there, and I'm happy to take questions.